Topic discussion on Department of Biochemistry. Dr. M.D. Mejbahul Islam, Lecturer, Adept of Biochemistry, TMSS Medical College, Bogura. After the end of today's session, student will able to define glycolysis, describe the steps of glycolysis, calculate calorie, generate in aerobic and anaerobic glycolysis, enumerate the importance of aerobic and anaerobic glycolysis, intermediary metabolism of carbohydrate, catabolic glycolysis. Mejbahul pathway converts glucose into pyruvate and lactate, producing energy. Citric acid cycle, Krebs or TCA cycle, oxidizes acetyl-CoA to CO2, serving as a central metabolic pathway for carbohydrates, fats, and amino acids. Glycogenolysis breaks down glycogen into glucose. Hexose monophosphate shunt, pentose phosphate pathway, an alternative route to glycolysis, involved in producing nucleotides and other molecules. Galactose metabolism involves converting galactose to glucose and synthesizing lactose. Fructose metabolism, the process of oxidizing fructose to pyruvate and understanding its relationship with glucose metabolism. Anabolic gluconeogenesis forms glucose from non-carbohydrate sources like amino acids and glycerol. Glycogenesis builds glycogen from glucose for storage. Amino sugar and mucopolysaccharide metabolism focuses on synthesizing amino sugars and other sugars for forming mucopolysaccharides and glycoproteins, which are important in structural and functional roles in the body. Glucose transport and its regulation. Glucose concentration in cells. Glucose levels are much lower inside cells compared to plasma, less than 100 mg/dl in humans. Insulin-independent transport system. This is a carrier-mediated process that doesn't require insulin, and it occurs in cells like hepatocytes. Insulin-dependent transport system requires insulin for glucose entry, primarily seen in muscle and adipose tissue. Glucose transporters, GLUTs. There are at least 6 glucose transporters, GLUT1 to GLUT5, and GLUT7, each with tissue specificity. For example, GLUT1, found in erythrocytes. GLUT4, found in skeletal muscle and adipose tissue, and insulin increases both the number and activity of GLUT4 in these tissues. Insulin resistance in type 2 diabetes. In individuals with type 2 diabetes, there is reduced GLUT4 expression in muscle and adipose tissue due to insulin resistance, impairing glucose uptake. Define glycolysis. Glycolysis is defined as the sequence of reactions converting glucose, or glycogen, to pyruvate or lactate, with the production of ATP. Salient features of glycolysis. Location. Glycolysis occurs in the cytoplasm of all cells. Oxygen requirements. It can occur both anaerobically, producing lactate, and aerobically, producing pyruvate, which is further oxidized to CO2 and H2O. Anaerobic glycolysis reaction. Glucose plus 2 ADP plus 2 Pi2 lactate plus 2 ATP. Branching pathway. Glycolysis intermediates are used for synthesizing amino acids and fats. Gluconeogenesis. Glycolysis can be reversed, with adjustments at irreversible steps, to synthesize glucose. Summary. Glycolysis is essential for energy production, especially in tissues with limited or no mitochondria, and its intermediates are integral to other metabolic processes. Reactions of glycolysis are energy investment phase or priming stage, B splitting phase, C energy generation phase. Energy investment phase. Glucose phosphorylation. Glucose is converted to glucose 6-phosphate by the enzymes hexokinase or glucokinase, isoenzymes. Hexokinase is present in most tissues. It can phosphorylate various hexoses, like fructose, mannose. Glucokinase is found in the liver, only phosphorylates glucose. Glucose 6-phosphate. Glucose 6-phosphate is impermeable to the cell membrane and has several metabolic fates, including glycolysis, glycogenesis, gluconeogenesis, and the pentose phosphate pathway. Isomerization to fructose 6-phosphate. Glucose 6-phosphate is converted into fructose 6-phosphate by the enzyme phosphohexose isomerase, requiring Mg2+ phosphorylation to fructose 1,6-bisphosphate. Fructose 6-phosphate is phosphorylated to fructose 1,6-bisphosphate by phosphofructokinase, PFK, a key irreversible step in glycolysis and a major regulatory point. Splitting phase. Fructose 1,6-bisphosphate is split into two 3-carbon compounds, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by the enzyme aldolase. Phosphoreose isomerase converts dihydroxyacetone phosphate into another molecule of glyceraldehyde 3-phosphate, resulting in two molecules of glyceraldehyde 3-phosphate from one glucose molecule. Energy generation phase. Glyceraldehyde 3-phosphate dehydrogenase converts glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, producing NAD and a high-energy compound. Phosphoglycerate kinase converts 1,3-bisphosphoglycerate to 3-phosphoglycerate, generating ATP through substrate-level phosphorylation. Phosphoglycerate mutase converts 3-phosphoglycerate to 2-phosphoglycerate, isomerization. Enolase forms phosphenyl pyruvate from 2-phosphoglycerate, inhibited by fluoride. Pyruvate kinase transfers a phosphate from phosphenyl pyruvate to ADP, forming ATP in an irreversible substrate-level phosphorylation step. Clinical importance of enolase inhibition. Fluoride is added to blood samples in the lab to prevent glycolysis, ensuring accurate glucose measurement. Dental caries is caused by bacterial infections, for example, Streptococcus mutans, that produce organic acids from fermentable sugars, eroding tooth enamel. Fluoride inhibits bacterial glycolysis and helps form fluoropatite, making teeth more resistant to decay and demineralization. Rappaport-Lubering cycle. The Rappaport-Lubering cycle is a pathway in erythrocytes that produces 2,3-bisphosphoglycerate, 2,3-BPG, from 1,3-bisphosphoglycerate, 1,3-BPG. Significance of 2,3-BPG, it allows glycolysis to proceed without ATP production, acting as a shunt pathway. 2,3-BPG binds to hemoglobin, reducing its oxygen affinity and promoting oxygen release to tissues. Its levels increase in hypoxic conditions, high altitudes, and fetal tissues. Anaerobic glycolysis. Pyruvate is converted to lactate by lactate dehydrogenase, regenerating NAD and to sustain glycolysis and ATP production. Importance. This is crucial in tissues like skeletal muscle during exercise and erythrocytes, which lack mitochondria, as well as in other glycolysis-dependent tissues like the brain and retina. Importance of anaerobic glycolysis. Energy in anaerobic conditions. Anaerobic glycolysis generates a small amount of ATP, 2 ATP per glucose, to sustain basic cellular functions like the NAK pump, especially during oxygen deprivation. Survival of glucose using tissues. Tissues like skeletal muscle that rely on glucose can survive anoxia, lack of oxygen, by continuing anaerobic glycolysis. In contrast, tissues that primarily use fatty acids, for example, cardiac muscle, cannot survive anoxic conditions. Generation of ATP in glycolysis. Aerobic, pathway enzyme, method of ATP synthesis, number of ATP synthesized. Glycolysis. Glyceraldehyde 3-phosphate dehydrogenase 6, 5, 2 NAD, etc. Oxidative phosphorylation, phosphoglycerate kinase, substrate level 2 phosphorylation, pyruvate kinase, substrate level 2 phosphorylation, Hexokinase and Phosphofructokinase, ATP-2 consumed, net ATP synthesis, aerobic. 8. 7. Generation of ATP in glycolysis, anaerobic, pathway enzyme, method of ATP synthesis, number of ATP synthesized glycolysis glyceraldehyde 3-phosphate dehydrogenase no ATP, oxidative phosphorylation, phosphoglycerate kinase, substrate level 2 phosphorylation, pyruvate kinase, substrate level 2 phosphorylation, hexokinase and phosphofructokinase, ATP-2 consumed, net ATP synthesis, aerobic. 2 irreversible steps in glycolysis, hexokinase, or glucokinase glucose and ATP glucose 6-phosphate plus ADP phosphofructokinase, PFK fructose 6-phosphate and ATP fructose 1,6 bisphosphate plus ADP pyruvate kinase, phosphenyl pyruvate plus ADP pyruvate and ATP regulation hexokinase /glucokinase, phosphofructokinase, PFK, and pyruvate kinase regulate glycolysis through their irreversible reactions. Hexokinase is inhibited by glucose 6-phosphate to prevent product accumulation. Glucokinase, specific to glucose, is induced by high glucose levels and insulin. Regulation PFK is the key regulatory enzyme, controlling the rate-limiting step in glycolysis. It is regulated by allosteric effectors, ATP, citrate, and H+, low pH, inhibit it, while fructose 2,6 bisphosphate, ADP, AMP, and Pi activate it. Fructose 2,6 bisphosphate plays a crucial role in activating PFK, thereby promoting glycolysis. Diseases linked to glycolysis issues, lactic acidosis. Prolonged anaerobic glycolysis leads to lactate buildup, causing lactic acidosis. Hemolytic anemia, genetic defects in glycolytic enzymes, for example, hexokinase, PFK, pyruvate kinase, impair RBC energy production, leading to cell destruction and anemia. Significance of glycolysis: it is the main pathway for glucose oxidation to produce ATP in all cells. It allows glucose, fructose, and galactose metabolism. It's essential for ATP production in anaerobic conditions, supporting survival during hypoxia/anoxia. In RBCs, glycolysis provides 2,3 BPG, crucial for oxygen transport by hemoglobin, and ATP to maintain RBC shape. Significance of glycolysis: glycolysis also provides biosynthetic precursors, 1-glyceraldehyde 3-P4-glycerol and tag synthesis, 2-pyruvate for alanine production, 3-glucose 6-P4-ribose synthesis, via HMP, 4-phosphoglycerate for serine synthesis. Thank you.